Hello everyone, my name is Krzysztof Kowal. I'm a doctor at the University of Life Sciences in Dublin, and today my presentation will be about my journey in bioinformatics field when I created the first tool. But first of all, I would like to thank the bioinformatics community for having us and for hosting us today. This is the first time when our city, our university is attending such meeting, so we are very glad to be a part of the bioinformatics community here with us together. I know that it was a long day, so I will do my presentation very shortly. So let's dive into it. First of all, I would like to point out that this Canis mitoSNP tool was created back then when I was a PhD student. Are there any students or PhD students in this aula who don't know how to code? Raise your hand, please. There are any people who don't know anything about like coding? Okay, so it's irrelevant. First of all, the background of the story. Okay, so first of all, the mitochondrial genome. It is a tiny genome which is just 60,000 base pairs long and encodes just 37 genes. And during my PhD, my goal was to analyze what were the changes in the mitochondrial genome in the comparison with the healthy tissues in cancer tissues. So the main goal was to analyze what were the changes that occurred during the carcinogenesis in dogs. Because dogs are a very good model for example, some human diseases. For example, mammary gland tumor is very similar to mammary breast cancer in females. So that's why it is very interesting field of study and also very good way to analyze what's happening in both species. So my goal during my PhD was to analyze what were the changes in the genome. So therefore, after the sequencing, we obtained the sequences, and as a PhD student that has a master's degree in biotechnology, I didn't have many skills linked to the bioinformatic analysis like coding or stuff. So therefore, I had to do all the analysis like with some tools like web applications or just the tools that were available online to perform the analysis. I didn't have these strong coding skills at the beginning, so therefore it was a little bit challenging, but it was just also very interesting to dive in this field of bioinformatics as well. So the mitochondrial genome is, as I previously said, a very tiny molecule in comparison to the nuclear genome. It's just 60,000 base pairs that are which encodes 37 genes. So we know that when we found some mutations in some protein coding genes or tRNA genes, it has to be done, the analysis of the changes and the effect of these changes on the, for example, secondary structure had to be done for each gene separately. So now, as I already am more skilled in bioinformatics and coding, I would do it in another way. But at the beginning, I had to download. I had these sequences. After the sequencing, I had to do the multiple sequence alignment, find variant calls that were mutations, polymorphism that occurred in these samples, and then I had to do the analysis of secondary structure of proteins and tRNAs in different types of tools. So it was just time-consuming and very hard to do work at the beginning, because each gene had to be done separately I had to translate the protein coding genes to the amino acid sequences and so on. So the analysis was quite demanding, especially when we had several samples. So then I came up with an idea how to speed up work, because if you are on PhD studies, you know that first and second year it's just maybe a little bit easy going, then it's a little bit harder and harder, and once you are closer to the end of the PhD, you know that you have to keep up with publications and so on in order to finish your PhD on time. So I knew that I have to do something which would be faster to analyze all the data that I had. And if I did it in 5 separate tools, I thought maybe maybe it would be easier to do one database to analyze all the sequences. So I know when I have like position, one mutation in genomic position, what would be the outcome for protein sequences, tRNA sequences, without extracting all the data, without separating the data, without cutting the sequences in order to analyze all that, just piece by piece. So this was the idea. And as I previously mentioned, I didn't have any skills, bioinformatics skills like coding, to do so. So I did all the data analysis, all the database in Excel, and that was the beginnings of my work. So I created the whole database in which I had just a genomic position, and I annotated what was the sequence from the reference sequence from NCBI, what was the complementary strand, that this position encodes one of the protein, what was the protein, what was the gene, then what's the position in the gene, what was the amino acid sequence, of course, and what the position was in the codon, and what the codon was. So I had the amino acid position for all the reference sequences. And it was easier for me to know what positions were in fact pathogenic, what positions were strictly mutated and had an impact on the secondary structure for the proteins, for example, because protein coding genes, proteins in mitochondrial genome, are responsible for oxidative phosphorylation process. So the proteins are in the membrane of the mitochondrial genome of the mitochondria. So the proteins can be inside of the mitochondria, inside of the membrane, or outside of the mitochondria. So I had all in my database annotated whether such position, what's the position in amino acid sequence, what's the amino acid, and then what was the part of the protein itself, and where it was located from other tools. And it helped me to have to fasten up the work from 3 or 4 weeks to just 1 day, just to have just a report of the sequences of the positions that were impacted, and then I could separate those that were irrelevant from those that were actually impacting on the proteins. So it was a lot easier for me back then. So why am I telling you that? Because generally, once you are starting your career in bioinformatics, or you are a biologist or biotechnologist or bioengineering from bioengineering studies, and you maybe don't have such skills, such developed skills, sometimes it's good to start with something that you already know or you're feeling good at. So sometimes to create something meaningful, something useful, reliable, it's good to start with something that will help you out. And then if it's working, this automatization of your work might be beneficial for others. Therefore, I wanted to publish my database. It was just one Excel file, honestly, and so it was useful even for today for many members of my team, and for others who are interested in analysis of canine, and both canine and human genomes as well. So that was the main goal of Canis mitoSNP. And as I previously mentioned at the beginning, I also wanted to analyze the comparison, whether if this position occurs in canine genome, whether this position is similar in human genome. So in order to have this comparative analysis, I compared all the sequences in human genome and canine mitochondrial genome. So from all these tools that were available, like for example Conserve, to know if the position, of the amino acid in the secondary structure is conservative or maybe variable. The information, what's the topology of the proteins encoded in the mitochondrial genome, and also what was the structure, secondary structure in the tRNA genes, and also what was the secondary structure or rRNA genes, and also the prediction method for the secondary structure of proteins were the information that I took for the Canis mitoSNP database. So the tool we created on website, we posted on website this Excel file. Maybe it wasn't very high, complex, and this tool wasn't very well sufficient and very well developed, but it was just worth it to do so to share with other people who might be able to want to do it also in a very efficient way. About the position 13,594, which is actually in both genes, in two genes, ND6 and ND5. So it happens in the mitochondrial genome that one position is encoding two genes. So we know that these positions are quite different. So if something impacts on this position, then in this position, we have an effect on two genes actually. So two subunits of the AGH gene. So in case of human genome, there is no such position. So it's something that it's happening just in canines. And in case of humans, there is a position in ND6 gene. So this is the basic, this is something that we can do the screening of the position of the sequences that are changed in case of canine or human genome, as what we prefer. So this is a good tool for the screening. So that was the presentation of a tool. In conclusion, as I told you previously, in bioinformatics field, sometimes it's good to just start to do something. So if you start the analysis, you have some data, you will think about how to do it more rapidly, more automatically, so you don't have to spend so much time in order to do so. Now we have also the programming skills. After I defended my thesis, I also went to postgraduate studies in bioinformatics. I learned coding, and now I would do it in a different way. But that was just the basics. Like it didn't need some high quality skills. It was just a simple Excel analysis, so just putting data. Now I would do it in SQL obviously, but as I previously mentioned, that was what I had as of now. So I want to tell you that don't be afraid of doing something for the first time, even if you feel that you don't have such high skills or something, that maybe you don't know how to do something. It can always be developed, it can always be done later, you can always learn something new, but the main idea is just to have a goal, have an aim to do something and start doing it. And at the end, I would just like to point out that as a member of Polish Committee of Genetics and also Bioinformatics, I'm in both societies, for Polish Society for Bioinformatics and also for Genetics. Next year, on the in September 21st to 24th, 2026 in Wrocław, there will be a conference, Polish Congress of Genetics, where there will be for the first time the section for bioinformatics. So if you are young students, young PhD, young scientists, you can save the date to present your work, your first things, your first presentation, your posters, your job that you have been doing, just to start to demonstrate yourself, and also to have contacts with others, with scientists working in genetics. I think it could be a good opportunity for you as young researchers. So that's all. Thank you very much for the presentation. Thank you very much for having me. If you have any questions, do not hesitate asking.